All right, welcome everybody to the January 21st edition of the New Jersey Mycological Association's Taxonomy Tuesday. Um, this evening, we're going to start off with a presentation on calicioid fungi, followed by some member finds of lichens. So I'll jump right into my presentation. All right, so calicioid fungi, sometimes referred to as pin lichens or stubble lichens. These are two photos that show the small black stalks with usually a capitulum of spores. Pictured here are Canotheca bruniola and Canotheca hygrophila. Both of these photos taken from the Delmarva manual by James Lundemer and Noel. So calicioid lichens are given the name calicioid because many of them were started in the genus Calicium, which is a genus described by Persoon in 1794. Persoon was born in South Africa, moved to Europe. Uh, he rented the upper floor of a house in a poor part of town. He was apparently unemployed, unmarried, poverty stricken, and a recluse. But he authored over 2,200 species of fungi. He was particularly prolific with describing polypores, but also described other species, including lichens. And he is credited with introducing the term lorella for the furrowed ascomata of the lichen genus Graphis. Since then, a lot of the Calicium species have been broken up into other genera. We have 7 genera of calicioid lichens known from New Jersey, including some in the original genus Calicium, basing that off of the 2019 publication by Dennis Waters and James Lundemer on the revised checklist of New Jersey lichens. So the full list on the left shows the 15 species that are confirmed from New Jersey, ranked by New Jersey rarity rank that I have assigned based on New Jersey records and habitats. Um, and then I also have on the bottom what I call bolo species. These are be on the lookout. So species that might be in New Jersey, but have yet to be documented. But especially when you're dealing with a group of fungi this small, it's easy to overlook them. So the fact that species have not yet been recorded from New Jersey does not mean they are definitively not here. So we're going to go through this list. I'm going to start off with the most common calicioid fungus, that is Phaeocalicium polyporium. Uh, Pheo from the Greek dusky or gray, and then polyporium because it grows on polypores, specifically Tricaptum biforme, although it has also been recorded from Tributes versicolor. This is a non-lichenized calicioid fungus. It is parasitic because it grows on living Tricaptum. Sometimes it's also called fairy pins. and can be distinguished from other Phaeocalicium species by their spores. This is a very abundant species. If you find Tricheptum biforme, you are likely to find Phaeocalicium polyporium growing on it. I frequently find this species on foray tables from people who have collected Tricheptum and unknowingly also collected Phaeocalicium polyporium. Probably about half of the fruiting fruitings of Phaeocalicium that I see on a log or a snag will have some with this calicioid fungus on it. It was originally described by William Nylander under the genus Calicium as Calicium polyporium in 1875. Wasn't until 1970 that Alexander Friedrich Wolfgang Schmidt created the genus Phaeocalicium. And then 9 years later, in 1979, Leif Thibault moved it from Calicium into Phaeocalicium. William Nylander was a Finnish botanist who taught at the University of Helsinki in the 1800s. He's credited with pioneering the technique of using chemical reagents to classify and identify fungi, specifically lichens. It's also the first to realize the effect of air pollution on lichen populations. And he described over 3,700 species during his career. Thibault is still alive, as far as I can tell. He's a lichenologist, also Scandinavian over in Sweden. He's a particularly proficient in calicioid lichens. We're going to see his name come up later. I couldn't find much on Alexander Friedrich Wolfgang Schmidt, but he's a German lichenologist, I believe. The second most common calicioid lichen that you're, or calicioid fungus that you're likely to come across in New Jersey is Mycocalicium subtili, Latin for subtilis, meaning fine, thin, slender, or delicate. This is also a non-lichenized species that grows on dry wood and bark, including pitch pine and the pine barrens. Here is the iNaturalist distribution of that species. You can see it's kind of got a northern distribution in eastern North America, but then it's also got this West Coast disjunct population. This is a species that, again, was first described by Persoon in the genus Calicium in the 1700s. Wasn't until Johann Reinke, a German botanist, created the genus Mycocalicium in 1895. Johann Reinke was credited for using the word Ceralia, which is the structure on the lichen that produces Ceridia, the propagule, asexual propagules that break off and allow the lichen to create a clone. And then in 1925, Udon Zatala, a Hungarian lichenologist, moved Calicium subtili to this new genus Mycocalicium. It's not a coincidence, I don't think, that the two most common calicioid fungi in New Jersey are both non-lichenized. But this species, Canothecopsis debilis, is what I believe to be the most common lichenized calicioid in New Jersey. We've given it a rank of S4, meaning it's uncommon but not rare. A Greek for to split or to gape, probably because it's the splitting of calicium, I think, yes. Then double A is weak or feeble. This is a more northern distributed species, but one that Dennis Waters has found fairly frequently in his recent surveys of Hunterdon County. It's a species that grows on the dry wood of standing hardwood snags, occasionally conifers, but mostly hardwoods. This is a species that was originally described by James Edward Smith as Lichen debilis, back when uh, most of the lichens were in the genus Lichen. Uh, Edward August Vanio created the genus Canathecopsis in 1927 until Thibault moved it into the present day genus in 1975. James Edward Smith was an English botanist and founder of the Linnaean Society of London. His library and botanical collections acquired European fame, and he was married to Pleasance Reeve, who survived him by 49 years, who eventually died at the age of 103. Edvard was a Finnish lichenologist, a student of William Nylander. He was extremely obstinate and was dedicated to his work. We find working at all hours, even on public holidays. Other biologists referred to Vainio's lighthouse, as lamplight would often be seen emerging, often well past midnight, from the windows of his small room in the older university building. He described more than 1,900 species. We'll see his name come up again in a little bit too. Next species, Canotheca hygrophila. This is a species that was described by Thibault. This is probably the most common rare species in New Jersey. I've given it a rank of S3. Notice the leprous thallus that the pins emerge from. This is a species that grows fairly reliably in higher quality Atlantic white cedar forests in the New Jersey Pine Barrens. Very few records on iNaturalist, but the herbarium collections show multiple populations from Bill Buck in the 90s and early 2000s, and then James Lindemer later on. Canotheca is a genus that was described by Theodore Magnus Fries, who was a Swedish botanist, lichenologist, and Arctic explorer. His father, the renowned mycologist Elias Magnus Fries, was sometimes referred to as the co-father of modern mycological classification alongside Persoon. Fries supported the Italian-German school of thought regarding lichen taxonomy, prioritizing fruiting bodies and spores in classification while downplaying thallus characteristics. Tensions escalated following Fries's 1861 publication where he criticized Nylander's classification methods. During Fries's candidacy for a position at Uppsala University, Nylander's objections were unsuccessfully leveraged to thwart his appointment. The Fries-Nylander taxonomic debate had enduring consequences for lichenology that persisted for decades. Eventually, Fries's methodology retained influential advocates, including Edvard Vainio. Also, something that I'm calling an S3 in New Jersey, Canathecopsis savanica, named after the Savo region in Finland. This is a species that grows on the bark and rotting logs of conifers and hardwoods. Similar distribution to other pin lichens, common in the northern latitudes. Few records from New Jersey. Again, mostly in the Pine Barrens, if not exclusively in the Pine Barrens. This is a species that was originally described as Mycocalicium savanicum by Veli Johann Paavo Bartholomeus Rosonen. Probably butchered that. Eventually, Thibault moved it to Canathecopsis in 1984, but Rosen was a Finnish lichenologist, apparently apprenticed with Edvard Vainio. You can see a common trend here of Scandinavian lichenologists. So for the remainder of these species, we're just going to kind of run through them a little bit more quickly here so that we are not here all evening. Gylidiopsis buckii, named in honor of Bill Buck, who's a bryologist and lichenologist who spent his career at the New York Botanic Gardens. This is a species that grows on small twigs and branches in sunny locations, either on forest edges or in wetlands. It's most often found on branches that have fallen from the canopy. It's more of a southern species that reaches its northern limit in New Jersey. A single herbarium collection from Dennis Waters from Mercer County. Although Joaquin Goodbar discovered a population of this down in the Pine Barrens. I think it was last year. This one was originally described as Tricharia vesdae by Bill Buck in 1980. But Vesda named in honor of Tony Vesda, a Czech lichenologist who taught botany at the Czech University of Life Sciences. He eventually got a job working out of his apartment, which also served as an office and herbarium. He had a collection of 10,000 reprints and 500 books about lichens, including 300,000 specimens that he kept in his home office. Vesda described this genus Gylidiopsis back in 1972, 8 years before Bill Buck described this species as Tricharia vesdae. But in 2005, these three authors, including Vesda, said, no, this really belongs in Gylidiopsis, and pulled an Uno reversal card and named it after Bill Buck. Same genus, Gylidiopsis mudia. This is a species that was described back in 2004. The type locality is in New Jersey. At the time, it was the only known location for this species in the world. Since then, we've found it widely distributed across Eastern North America. This is a lichenized species that grows on bryophytes and organic matter in early successional habitats, such as power line rights-of-way, roadsides, open fields, and the roadbeds of gravel roads. It says in the Delmarva Lichens book that the only way to find Gylidiopsis mudia is to get down on your hands and knees in a habitat where you are likely to get chiggers, and then search the tiny tufts of dead grass and organic matter for signs of a thin film and reddish-brown hyphophores. So we only have one known location, the type locality for the species in New Jersey, and hasn't been found anywhere else in the state since then. But since it's a small nondescript species in a fairly common habitat, I gave it a rank of S2. Suspect there are populations out there in New Jersey that we have yet to find. Here's a map of the distribution in Eastern North America that have been found since it was first described in 2004. Got one more species of Gylidiopsis. This is Bartramiorium. which has been found down in the Pine Barrens. Again, this, like buckeye, is a southern species that reaches its northern limit in New Jersey. Calicium lenticulare is an S1.1 species, meaning it's only ever been found at a single site in New Jersey by Bill Buck. And it was found in the Bear Swamp in Cumberland County, which is widely regarded as our best, if not only, true old growth primeval forest in New Jersey. You can see it's got somewhat of a widespread distribution along both the east and west coast of New Jersey, of the United States. Here's another S1.1, this one collected by R.C. Harris, also from Bear Swamp East, Canatheca xylosina. Also has both an east and west coast affinity, not so much in the middle, except for some areas in the upper Midwest. Calicium abitinum is a species that was found by Austin, apparently, back in the 1800s in Bergen County, but then James Lendemer found it in the Pine Barrens, Burlington County in 2003. Has a pretty widespread distribution in North America, but we only have a single locality for it in the state, so I'm calling it S1. Another Calicium adspersum. This is a pursoon species. Was collected by C.F. Austin, again, back in the 1800s. But hasn't been seen in New Jersey since, so I'm calling this SH. Another historical species, Canatheca phaeocephala, was included on the checklist for some 1800 records, but I couldn't find a precise specimen. So again, ranked SH. Mycocalicium fusipes. This is a species I couldn't find any photo of at all, even though it has been collected quite a few times in the 1800s, but hasn't been found in New Jersey since. It has a pretty dispersed distribution in North America. Stenocype, another historical species, found in the 1800s, most recently by J.B. Ellis from Gloucester, likely in Newfield. This is a species that apparently grows only on the branches and bark of Alnus species, Alnus being the plant genus for alders. But again, not being seen since the 1800s doesn't really mean much when you're dealing with the species so small and inconspicuous, and which appears to grow on such a specific substrate. So then we've got these bolo species like Calicium parvum. This is a species that grows on the bark of conifers. This one, for some reason, was excluded from the checklist. If you look at the map there is a dot in New Jersey, roughly over the Pine Barrens. This was, I believe, collected by James Lendemer from the Pine Barrens, but just before the New Jersey checklist was published. So I haven't added any species to the checklist until it's been peer reviewed and an addendum to the checklist has been published. So for now, this is a bolo, but chances are it's here. And if an updated checklist is published by James and Dennis, I'm guessing the species is going to be on it. Here's another species, Calicium salicinum. which grows on old willows or old oaks, which is where it gets its name. The genus for willows is Salix. This is a species that grows in the Delmarva. It's in the Delmarva book, um, and has a pretty widespread distribution in Eastern North America. So presumably it's could very well be in New Jersey. Here's Calicium trabinellum. This one seems to be a northern species, kind of this clear geographical distribution where you've got this northern affinity with that Appalachian spur kind of going down the Appalachians, or a disjunct set of populations in the Great Smokies. This one's kind of interesting because the authority here says ACH in parentheses and then ACH. So forgetting the botanist who ACH is an abbreviation of, but he originally described this in 1803 as Calicium xylonellum variety trabinellum. And then 7 years later, in 1810, he updated his own authorship and elevated it to a species level. You can also see here that James Edward Smith described this species as Lichen trabinellus, but that was in 1806. So because that postdates the 1803 record, this is technically the first described account of the species, and gets the credits in the parentheses, which is why this record is in parentheses, not the Smith record. Here's another bolo species, Canotheca balsam conensis. This is a species that was just described back in 2015. It grows on Tricaptum abitinum, and it's a lichenized pin. So if you recall, the most common calicioid species in New Jersey is Phaeocalicium polyporium, which grows on Tricaptum biforme. So this is a species that could very well be being misidentified as Phaeocalicium polyporium if people see that it's growing on Tricaptum and just assume that it's the common non-lichenized Phaeocalicium. But in the article that describes the species, Jesse Allen and McMullen say, we suspect that the species has a broader distribution that closely follows the distribution of Tricaptum abitinum. We certainly have that Tricaptum species in New Jersey, so this is something that we should be keeping an eye out for. Canotheca bruniola. It's another species that widespread in Eastern North America. Technically hasn't been documented from New Jersey. You can see this has sort of a light green leprous thallus with these stalks that shoot up with a golden capitulum. Another Canotheca chrysocephala, this one with more of a yellowish green thallus. This one looks to be more of a higher elevation Appalachian Mountains distributed species that grows on mostly conifers, particularly spruce and larch, sometimes pine. Canotheca ferruginea. Took me a while to even find it in this photo, but it's right here in the middle. These black pins here. This is the best photo I can find of it. This one's a pretty unique looking one, Canotheca furfuracea. These yellowish green leprous thallus and these long, skinny stalks. This one grows on soil or on roots protected from precipitation. There seems to be a a common theme. These things don't like to get rained on. They presumably like humid environments, but they don't like to get saturated and super wet. This is a species that was described back in 2021, Cryptocalicium blascoii, described from collections in Spain. It's a monotypic genus. They created the genus Cryptocalicium specifically for this species. In fact, not only did they describe a new genus, they also described a new family, a new order, and a new subclass, all for this one species that grows on the inside of peeling bark on trees in the family Cupressaceae. And then in 2023, it was found in North America, including in New York, where it was found growing on the inside of peeling bark on eastern red cedar, Juniperus virginiana. So you can see the arrows here in this photo labeled B, all these white carrots that are showing the inside of the peeling bark where this lichen, non-lichenized calicioid species can be found. They also say in the publishing article, it's probably the tiniest known calicioid fungus. And it's so tiny that it's can be hardly observed in nature, even using a hand lens. And it's best surveyed by gathering loose bark strips and placing them under the stereo microscope to see if you can find it. So again, not surprising that it took so long to describe, and now that it's been found in North America, is something to keep an eye out for. This is another species that has a very specific substrate. This is Phaeocalicium curtisii, that has a historic record from Ellis in Gloucester back in the 1800s. And then it was found very close, just over the border in Pennsylvania. But this is a species that grows exclusively on the bark and the base of dead stems of sumacs, especially staghorn sumacs. So if you find any staghorn sumacs that have dead or dying branches, take a close look at the surface to see if you find any calicioid species. So this is a publication that's free and available to download over the internet by E.B. Peterson. This is a key to the Calicioid genera. And you can see here some of the couplets that are used to split the various genera in North America. Size of the stalk, the color of the spore mass. Occasionally you'll use chemistry to see if there's a reaction. Usually chemical reagents with calicioid are observed using a compound microscope. But why do we care about these small, inconspicuous stubble lichens? Well, they've been used since the 1970s as indicators for habitat quality and identification or assessments of old growth forests, especially by Francis Rose, who in the 1970s started studying these things, especially in older growth forest or old growth forest of the UK. and then eventually developed an index of ecological continuity. Um, Here's a map showing various grids and the number of Calicioid species uh, having more of a reddish color. And those are basically where most of the old growth forests are located in the UK. And then in the 1990s, Stephen Selva up at the University of Maine uh, published a similar methodology for assessing old growth forests of New England and produced this list back in 1994 on species that appear to be faithful to ancient forest conditions in New England. Um, so he's done various inventories in old growth forests across New England, and eventually found that paying attention exclusively to calicioid species is more time efficient and accurate than using all these other lichens that are listed here. So eventually he's using calicioid lichens exclusively and has found that much like over in the UK, the number of species found is a highly accurate way of identifying old growth. Young pioneer forest, early successional forest, less than 40 years old, might have one or two species of calicioid fungi. An old growth stand could have over 20, sometimes over 30, and intermediate aged stands have an intermediate number of calicioid species. He's not particularly fond of using indicator species, oh, the presence of this one calicioid lichen means that this is old growth. He's more, he more supports the use of the total number, species richness, or total number of species that are found in a stand. And this has been, you know, widely adopted, broadly the use of lichens to try to identify or to justify designation of old growth forests, including designation of state natural areas or designating areas of protection from logging or to be, forest to be managed as old growth. Here's another paper by McMullen and Wiersma talking about the use of lichens as indicator species for identifying old growth forests. The New York Natural Heritage Program has a similar methodology called the OGRE protocol, the Old Growth Rapid Evaluation, that people can use to quantitatively assess whether or not a forest meets their definition of old growth. And even though they don't use calicioid lichens, probably because they're so difficult to find and identify, they do have 3 indicator species that they use in their calculations or assessment, including Lobaria pulmonaria, the lungwort lichen, as well as a species of moss and a species of liverwort. So even though these things are small and conspicuous, they can be pretty powerful tools in helping ecologists justify or identify designation of old growth forests, which can have management or conservation implications. So that's my presentation on calicioid fungi of New Jersey and their use and importance. Happy to take questions or move on to some member finds.
Jason, that was great. Yeah, that was amazing, man. Thank you.
I've got one question, and maybe it's kind of basic, but can you explain the difference what you refer to as lichenized and non-lichenized?
Right.
So is there such a thing as a non-lichenized lichen? It's kind of
Nope.
a
No.
okay.
So a non-lichenized fungus, well, a lichenized fungus is one that has partnered with a photobiont, either an algae or a cyanobacteria.
Mhm.
So you can determine if you have a specimen that you're unsure is lichenized or non-lichenized, you can, put it under a microscope and see if you see algae. Um, non-lichenized means that it's non-lichenized fungus is, one of the other types of fungi, and then either mycorrhizal, saprobic, or parasitic. So the non-lichenized calicioids, some of them like Phaeocalicium polyporium are parasitic as they colonize living Tricaptum biforme, and then some of them are saprobic, like the Cryptocalicium that grows on dead or dying bark.
Jason, did I hear that correctly? There are some mycorrhizal, non-lichenized um, fungi or non-lichenized calicioid.
No, there's no mycorrhizal calicioids, but mycorrhizal fungi are, one of the non-lichenized fungi.
Oh, okay. Oh, I see. I see. Um,
All
aside
right.
from oh, God.
Yeah, so just to clarify then, so some of the things that you showed us tonight um were calicioid fungi, but not necessarily lichens. So
Correct.
yeah, okay, Phaeocalicium, there are some that are lichens and some that are not.
Right. So the Phaeocalicium polyporium is non-lichenized.
Mhm. Okay. All right, so it's not getting its food from an algae or a bacteria, it's getting it from the living tricaptum.
Right.
Okay. All right, sorry, Dave, if I cut you off there.
That's all right. Um, aside from substrate, how does one go about trying to identify these things? I suppose microscopic traits, and in which case, how do you get the microscopic traits? Do you have to search for the spores by sectioning, or just scraping onto a slide? Or
I think smash mounts are probably the best way to go to find spores, and then doing the smash mount in different chemical reagents to check for um reactions. There are keys out there. I had the key um to the Calicioid genera, but the see if you guys can see this. You guys see this PDF, even though I'm still sharing
Yes,
my
yes.
presenting my slideshow. So this is the uh, um, lichens of the Great Smoky Mountains. So they have a whole separate key for calicioid fungi that um, goes through measuring the size of the stalk. What is the substrate, measuring the ascospores, the shape of the spores. So there's because this is the Great Smoky Mountains key, there's going to be a lot of species here that are not relevant to our area, but it gives you an idea of some of the traits that they use.
it sounds to me like to look for these, you really are searching for specific substrates to be able to examine.
Yeah, for some of them, um, a lot of these, like the Mycocalicium subtili. This is a species that I found a few weeks ago at Franklin Parker, and it was just growing on a standing, living pine tree. Certainly some of the rare ones have, highly specific substrates, but that's why they're, even the non-lichenized species are typically found or collected by lichenologists, because if you don't have your nose up in the bark of a tree, you're not likely to see it.
So when you say calicioid fungi, is that referring to the pin shaped ones? Because when I was coming into this, I'm looking up things in like Caliciaceae, right? Um, which they're wildly kind of variable things in that family. So when you say calicioid fungi, are you more or less referring to the pinhead type things and not necessarily things like Buellia and things like that, which are in that the family Calicium of Calicium?
Right. Yeah. So calicioid is, refers to species that uh, are in or closely resemble members of the genus Calicium. So just the pins.
Cool. And then also, you mentioned that they're going to, that was really interesting that they're indicators of old growth, and they're used to actually, there's a level of measurement there. But are these things growing on the old growth trees, or are they just present in old growth forests? So you can find them on twigs and young growth, newer growth, um, just in an older growth setting, right?
Yeah. So there's probably a few things involved in why uh, lichen diversity as a whole, including calicioid lichens, tends to be higher in older growth forests. One is older growth forests have older trees, and older trees have bark that have different chemistries, different textures. They tend to be gnarly and have more heterogeneity, so there's more microhabitat in older growth forests than you typically find in younger forests that have younger trees with relatively smoother bark. Older growth forests also um, tend to have uh, more epiphytes in general, whether it's mosses, lichens, you just have more propagules. It takes a long time for things to colonize an old growth forest. So um, when you have a forest that has a high abundance or cover of species on the bark, it acts as a buffer to humidity. So when it rains, those epiphytes absorb water and they hold on to it and they release it more slowly over time. So there's a younger forests that don't have all of those things covering the trees tend to dry out. The air within the forest dries out more quickly. Lichenologists have said, like James Lundemer and others have said that even in upland forests, most of the East Coast forests before they were originally cut down by colonists um, likely would have been covered. Every inch would have been covered with something, polypore, a fern, a moss, a lichen, something. So the uh, Pine Barrens is probably an exception because it's a fire adapted ecosystem, but the ability to identify trees by their bark is potentially a modern phenomenon that you wouldn't have been able to do, hundreds of years ago, because they would have been so covered in various epiphytes, which makes sense because there's a common adage in ecology: nature abhors a vacuum. Right? So if you have this this part of the bark on a trunk of a tree that nothing is colonized, then that's free real estate. It's something that something can colonize without any competition. So it doesn't make sense that there would be nothing growing there. And then the last reason is that these things disperse very slowly. Especially species that are reproduce sexually with spores that have to find a suitable substrate and then photobiont. The distance that those spores travel within the dense interior of a forest, can be pretty restricted. It's unlikely that a wind or storm event is going to pick up a spore and travel it miles. So it's possible that these spores are only dispersing in any abundant fashion, just a few feet at a time. When you couple that with the lack of old growth forest and any substrate restrictions that are required of older trees, and you can see that these things are going to be very slow to recolonize areas that were widely deforested in the 1800s and early 1900s, and remnant populations that have somehow persisted to today are of high conservation value because they're these remnant populations that are helping to recolonize other forests or are waiting in dormancy until the forest around them sufficiently matures so that they can start to recolonize areas.
Very cool. Thank you.
Did I hear you correctly in saying that with the lichenized ones, or I just assume lichens in general, you could look for the algal cells?
Yeah, so if you do a smash mount or section, I mean, probably not with calicioid lichens, but there's some species that um, if you're trying to figure out if it's a lichenized crustose or a non-lichenized crust, and if you just kind of slice it with a knife and look at it under a loop, if it's a lichenized crust, you should be able to see that layer of algae sandwiched in between the upper and lower cortex, or the within the medulla. And yeah, that is usually visible through a loop in the field. You can usually see that line of algae if you do a little quick dissection. But you
So
can
you're
also
looking for the different separations in color, really.
right, yeah.
All right.
But you could also see it under a compound scope. I mean, you can see the actual algal cells. If you notice in this key here, the Great Smokies, one of the couplets, couplet 21, has you identifying the photobiont. If it's an algae in the genus Tremtopolia, then it's one species, and if it's a trabuxioid or different type of algae, then you go down the rest of this key.
And now we got something new to look for.
Yeah, totally. I'm already going through previous observations like, oh, do I have anything that looked like what Jason
All right, any more questions on calicioids, or should we move on to member finds?
I still have some questions, but I'll ask them when I share some of those observations.
All right. So if you have anything you'd like to share, say so in the chat. I think I have some that were emailed to the Taxonomy Tuesday email we could bring up. Let's see. Virginia, you're here. You want to start us off?
Okay, all of mine are from Maine and they're not very recent because I took Steve Selva's course a couple times up there at Eagle Hill. So that's where most of where my calicioid lichens were all found. I think this was Stenosope major. And I think there might be, yeah, there's the spores. You identify it by the spores, a certain number of septa. And I think this was the one that was not lichenized. So I probably didn't find any algae in the base or anywhere. And these were found probably mostly around Eagle Hill or Great West Island. I don't remember specifically on this one.
Cool. Found on balsam fir.
Mostly, yeah, 3 septate.
And was the course, the Eagle Hill course, was it Perinomycetes or was it a calicioid fungi thing?
It was calicioid fungi. It was Steve Selva, and he taught the
cool.
course. Yeah, and then he's I think he's retired now, but he's from Fort Kent, the University of Maine up at Fort Kent was where his location. I guess he's still up in that area. But he taught the course several times at Eagle Hill. So as you can see, the similarities between these calicioid lichens and slime molds being very tiny. And basically that's how one thing led to another for me, because I was collecting the calicioid lichens and I found one that was not. It was a slime mold. So I went on from there. And I haven't really looked at the calicioid lichens for quite a few years now. And this is Chenotheca bruniola, and that was rather common up there in Maine. I'd collected a lot of these, but most of mine don't have good photos, so I couldn't put a lot on iNaturalist because you have to have a good photo. Okay, so that photo right there shows the algae at the base. There's a clump of algae. So it's not a very good picture, but that does help to identify it. And the Chenotheca has a brown, the mesothelium is that clump at the head of spores, the clump of spores. It's called a mesothelium. And they have spherical spores that are light brown. That's another characteristic. No septa of the Chenotheca bruniola.
Virginia, that a shot of microscopy is showing the algae. Is that what was the level of magnification for that?
Oh, I can't remember. This was several years ago. I probably
okay.
go up to 1,000 for most of them. I just, if I saw something, I would just crank it up to the next level. And you can see that's a clump of algae surrounded by like a thicker wall or something. And I don't remember the name of that particular algae. I used to know the names of some of them.
And do you remember
Dictyochloropsis? Is this the genus here?
What's the question?
Dictyochlorops, is that the name
Oh,
of the
yeah,
genus?
if I had that, that's what I identified it as at the time, which I don't
Yeah.
remember now anymore. When you don't use something for a long time, you forget. And I haven't looked at these for a long time. And I could be wrong on the algae because they're sort of difficult.
Yeah.
Mhm.
Yeah, and I was going to ask, do you remember if to the naked eye, I mean, obviously you saw something to take this photograph of the algae under the scope there, but do you remember if it was that green or if that green kind of processed in the photo? Because that's something that I've noticed. I won't notice anything green, and I'll take a photo of a spore, and I'll notice an algae spore floating around in there that, for whatever reason, shows up brighter with
Yeah, well,
photograph.
if I make a smash mount of the, I use a razor blade or something to pick it off the substrate and smash it and look at it under the microscope, and I'll look for the green algae cells around the base of it, or sometimes you can see if the substrate is greenish.
Cool. Okay. Thank you.
Cool. Thanks, Virginia.
Yes.
It seems like in a lot of the observations, the wood is heavily decayed. There's no bark on any of the substrates. Is that, do you think, just a product of that it's easier to find them in that situation, or it, because it sounded like in the old growth forest that wouldn't really be their preference. Or
Yeah, I don't know. I'd have to study the specific ones, but a lot of these are found on bare wood. The bark is gone, and it just looks like whiskers on bare wood, and that's one of the characteristics that you look for. And you
Yeah.
just have to use, if you're looking at the bark of a tree, just have to get your nose up to it and just look at it um very carefully, or between the bark. I don't remember the name of it, but there's one that you find, you sort of look at the ridges in the bark and try to look in between and see if you find something.
right, like that one from Spain was like that under the bark. Yeah, I was just wondering if it's just the, ease of visibility, that because not just your observations, but so many of the ones Jason showed were also on there. And so I'm just curious, like, oh, is that where I really want to be looking more, or is it just more a matter of ease of visibility?
You want to look there first, I guess, on these, an old snag or old wood that has all the bark off of it. Look at that closely. I mean, it's just like slime molds. You gotta bend down or look at things, way up close.
Mm-hmm.
Yeah, I think by the time a tree falls and becomes a log, it's going to get overrun with mosses, and the mosses are going to outcompete the calicioids. So yeah, I don't think it really matters too much whether or not the bark is dead or alive, but I think the fact that it's standing probably is preferable to fallen logs. I mean, there's certainly some calicioids that you could find on logs, but I would definitely be looking at standing snags first.
Right. I think that's one of the things that Steve Selva said to look at was standing snags.
Cool, thanks.
All right, thanks, Virginia.
I think there was one more. I don't know if it came through or not. I had trouble sending it.
Let's see. Oh, maybe. Is this it? Ah, okay. It didn't come up as a
Yeah,
this
that's
one.
it.
Cool.
Phaeocalicium. So the phaeocaliciums are rather specific, I think, to the trees that they grow on. Some of them grow on birches. I forget if this one might have been on a birch. I don't remember now. Specific,
Paper
yeah,
birch.
spatula, yes. They are rather specific on what they like to grow on. Like the other one that was mentioned before, Curtisii on
Sumacs.
Sumacs, yeah. Curtisii. I know one of these Steve named after his son. I forget if it was the Curtisii or this one, Matthewsianum.
Cool. Now, what's this red here at the base? Is this
That was
mounted in
probably a
something?
piece of the bark. Could
Okay.
have been a piece of the bark. And I don't remember if this one was lichenized or not. And there's the asci or the mesoidium, whatever, with rows of spores in them. And then those spores were septate, I think. I think most of the Phaeocaliciums might be septate. I think that was from Great Lost Island, probably. Mhm.
Is there a preferable liquid to mount these smashes in to find these spores?
I guess just use water, but you do need some chemicals to identify specific species. The Calicium, and I forget now the chemicals,
Like
iodine
IKI.
maybe, some of the Caliciums, and then there's another red dye or something you use to identify some others, but it's been so long since I've worked with them that I don't remember exactly
see.
what you use. You do need some chemicals.
But it looks like the spore morphology, the details are coming through pretty well.
Yes, I think
see
they
that now.
do. You're right.
This is the paper that describes the Cryptoculicium. There's a
Yeah, that's a new one to me. I never heard of that one before.
section here. Yeah, there's a whole ascospore shape, size. It gets into this whole amyloid and KOH plus IKI. I think IKI is iodine. Forget what IKI is. Surface of the stipe and subhymeneal dark violet opaque, partly dissolving and turning turquoise green in KOH. So yeah, sometimes finding the paper that originally described the species, especially for things that were described more recently, is going to give you a lot of details to help you identify things that are not in a lot of the treatments or keys. All right.
Okay.
Dave, you want to go next? Dave, you still there? Maybe Dave stepped away. Anybody else have anything they want to share? Come back to Dave. Brandon, you want to go next?
Sure. Yeah, I can do that. You want to stop your share?
Yeah. Yeah.
All right. All right. Can you guys see my screen?
Yep.
All right. So I was scrambling as you were doing that. Really cool. I appreciate you doing a little presentation on these things. They're really cool. So I found this a while ago, and when you showed that the one that was growing on alder, I think you said it was.
Right.
What was the name of that, by the way? I didn't get a chance to write it down.
That was the
Would there be stenosoby?
yeah, was that the stenosopy? Let's see. Yep, stenosopy pulatula. I'll put it in the chat.
Cool. And I literally I had no idea what I was looking at when I was looking at well, we'll go back, these Rosalinea here in the foreground, um, here under the stereoscope, and I found a couple just oddball random things, which it makes sense when you had mentioned sometimes that's the best way to look for these things is just find a high activity kind of piece of wood and search under the stereoscope. Um, but I thought it looked similar at least, although this seems to be pretty branched. Do you think that's a possibility here?
No, I think that's something else.
Okay.
It's branched, and the width of the stalk seems a little bit too wide to me.
Probably just some other non-lichenized ASCO, maybe.
Yeah.
All right, cool. And my other question was, so I was looking at this Orbilia when I found a bunch of just crazy ascomycetes and lichen under the scope. Um, do you think that this, if I sent this out or you commented on it, and I think we kind of both black birch was our first thought, but do you think this could be an alder? I do not know my trees.
No, it's too big for an alder. Alders are typically more shrubs.
Okay.
You would never find an alder that large. Alders
Okay.
also tend to grow in wetlands, and they tend to have more of a shrub habit.
Nice. Thank you. I really don't want to have to wait until this thing starts having leaves to figure out what it is, because I really want to figure out what that other thing was.
Well, if it is black birch, if you take a knife and kind of drill a little bit of a hole in the bark and smell it, it'll smell like birch, root beer or birch beer.
Oh, okay.
Yeah, you can even get that from twigs.
Yeah.
Okay. That's a good note. Thank you. And then this thing, I found this week, and I was photographing these cladonia. That they're out of focus, but I noticed these things I didn't notice originally, and they were very, very small. You can see one there, kind of these globules on a little stalk, and I thought that looked like one of the things that you showed that started with a G, Glylidiopsis. But I'm not sure how to distinguish if this is like a young growth cladonia apothecia, or if it potentially is that other Moodii, I think you called it, Moodii.
Yeah, I mean, it looks like it's coming out of the soil. I think the Glylidiopsis, tends to be attached, directly to detritus. It's hard to get a sense of the scale. I'm wondering if this is Debeus Beomyces. that as a question mark.
By the
in
way,
chat.
this was on bark.
Oh, okay. Well, that changes things. I thought it was on soil.
Yeah, but that was actually something that I was kind of pointing me against, because didn't you say that that Moutier thing grows on soil, or that's where you look for it?
Yeah.
Yeah, this was on bark with mosses and a cladonia. So, can you put the that species that it might be in the chat for me?
Yeah. I mean, the Jubaeus beomyces is typically on soil, in sand.
Yeah.
I don't know that it doesn't grow on bark, but trying to think, there's species.
Yeah, and that the one that you showed, not necessarily like it was pretty early on. The Gylidiopsis Moutier.
Mhm.
Um, was that a little bit atypical for these calicioid things? That seemed to have a little more color and not be as kind of carbonaceous as the rest of them look.
Yeah, I think that's true.
Okay, cool.
It's kind of a weird. I mean, it's almost stalkless. It grows in these weird habitats, so it's. I think it's a little bit more, less permanent, I guess you could say, than some of the other calicioids. It colonizes a habitat that tends to be, early successional, or more of a pioneer species than some of these other old growth type species.
Yeah, and that was what kind of rang true with this one. You mentioned that it or what I was proposing this could possibly be, because I found this at Black Run Preserve, which is definitely not old growth. Um, Maybe early successional, um, but yeah, that also fit. But whatever that thing is, it's cool, and I've never seen anything quite like it.
Yeah, you didn't get any like closer. I don't see like real up close.
No, no, I really didn't. Actually, I struggled to photograph these. They were, like I said, they were really really small, and look at there's this cladonia in the foreground. Like my focus was just didn't know where to go.
Mm.
But I didn't collect any either. I know exactly where it is, so I can go back. And they're often.
Were those leafy or were they more like nodules?
I would say more like nodules. What do you mean, like the
Yeah,
apothecia, like the cap?
what I was wondering is there might be like some peltigera mixed in there or something, which would be kind of like not leafy, but geez, I don't know how I would describe it.
Like lobate.
Lobate, yes, good.
I mean, there could be some peltigera in here. I've, I'm learning with these things. It's they're look at how many things are just in this photograph, right? I don't know if these if these squamules, right here at the base are part of this lichen, or if it's it looks more like a non-lichenized ascomycete, to my untrained eye, right? But let's move on. And then when I found this, maybe a couple weeks ago, yeah, January 8th, I think it's a lichen, and I called it Buellia, but I do not know the genus very well at all. But these cups were pretty tough, I guess. Yeah, like not quite carbonaceous, but they weren't like your typical ascomycete cup fungi type things where you could, they were almost jelly-like. These were much tougher. But again, I do not know what it is. I don't know if that thallus, or if that's a thallus or a subiculum. I guess I'm not even sure what the difference is when it comes to ascos. So yeah, and Jason, do you think this is a lichen or a non-lichenized asco?
It doesn't look like any lichen I know.
Yeah, you can see them there.
What are those little black ascos? I forget the name. It sounds like Bulgaria. Or there might be like a pseudo one also, or I'm just not coming to me now.
No, there's Bulgaria and Iquins, and I think there is something else. I don't. Let me search.
That one's much larger.
Yeah,
This is really tiny.
very tiny.
Yeah, you can see my.
Okay, yeah, these are too small.
You
Yeah,
can see my thumb
right.
there. The either subiculum or whether that actually is a different fungus, and these are like growing on a second fungus. It very much reminded me of a dried out and black version of what's that thing? Hydnoporia, Hydnoporia olivaceae.
Yeah, go back to that last picture. So this is why I'm thinking it's not a lichen, because this thing looks like it's colonizing the inner lignicolous part of the bark. So this isn't.
No, it was only on the bark. You can't see
Oh,
any.
because
You can't see any of the bark because the rest of it had peeled away. But I peeled a piece of bark. Like I have this on a piece of bark.
so
Yeah,
it's
see
not.
right here.
So what was this? Do you know what this was on? What species of, was this a tree or a?
So it was on a somewhere between a branch, yeah, like this size, maybe 3 inches, and I want to say it was white birch, paper birch.
Any silver birch? Silver birch
Yeah.
jumped to my mind. Is that around this area? Where you found it? I agree. It looks like some kind of birch.
But
Was
whatever
it planted?
whatever it was on, so this was on a man-made walkway
Okay.
through like a marshland, right?
Okay.
So just a bunch of random sticks that were laying around that someone created a walkway through a wetland. So
unlikely that it would have been planted, like the tree would have been planted.
correct. Oh yeah.
So it was a wild species.
Yep.
It's unlikely that it would be either paper or silver birch
Yeah.
if it was grown in the wild
Okay.
in Black Run Preserve. The only birch that I can think of that would be growing wild there would be black birch.
What about water birch?
You mean river birch?
River birch, I mean. Yes, I'm sorry. Right.
I don't think that would be Black Run either.
Although I gotta argue with that a little bit because less than 50 feet from here, this exact where I found this thing, there is a white birch. Well,
Yeah, gray birch.
Yeah, gray birch and white birch are easy to confuse. White birch peels quite a bit.
Yeah.
yeah, and would not Piptoporus, Fomitopsis betulina. Would that grows on both?
I
There's
would think
buckrow
so.
But that's the typical birch polypore, right?
Mhm.
right.
Yeah, that grows on all the birches,
All
I
of them.
think.
I think
Yeah,
so.
gray birch is another species that could be at Black Run.
Because I know, I definitely know how to identify Fomitopsis speculina, and I've found that less than 50 feet away from here. So could have even, and it's on a fallen, uh, a fallen, no, full fallen tree, and it's
Mm.
fully inoculated with this Piptoporus. So it could have been a branch from that tree
Yeah.
that somebody that is maintaining this area used. But I do think it's on birch. But again,
Yeah, it'd have to be gray birch.
gray birch.
But-
All right, that's a good lead.
Yeah, that
But back to your comment about the what was it, the Hypnoporia alivacea? It
Mhm.
kind of looks like something almost colonizing that. I can sort of talk myself into to seeing Hypnoporia there, but not quite.
Yeah, that was kind of exactly where I was with it. Whether or not it was this odd. What is the difference between a thallus and a subiculum? Does anyone know?
I don't know the term subiculum.
All right.
We'd have to look it up. Not my everyday word.
All right, we'll move on. Um,
So I just looked it up. Subiculum is a crust-like growth of mycelium under fruit bodies. So if the subiculum is made of mycelium, that would be different than a thallus, which is typically a at least with lichens.
membranous.
Yeah, it's like it's it's got the medulla with the
Yeah, it's got
cortex.
like multiple layers of
Right.
different things, whereas a subiculum would just be essentially a mat of mycelium that
Right.
might be a certain color or something like that. All right, cool. Wait, did I share this one already? No, I didn't.
No.
So I was calling this Buellia, a new stab at putting a name on a new lichen for me. But you can see these little mounds. They're not perfectly circular. They're kind of lumpy, kind of not arranged in any kind of way. On a pale thallus that's cracking a little bit. My very brief, dive into trying to identify this thing. Buellia, yeah, or however you say that, was my thought. But there's a couple things that also looked similar. So, what do you think?
Yeah, I think Buellia looks good. I mean, I am kind of more used to seeing Buellias with a kind of a flat top and not these kind of lumpy masses, but I still think it's a Buellia. Can't think of what else it would be.
The first thing that I looked at was that gosh, what's the name of that thing we found at one time? Basidia,
Basidia?
but not spelled like we're used to with a Basidia with a C.
Right.
But I don't think it's that, right? At least not Schwarnitzii, which
Not
is the
Schwanitziae, because the thallus was it wet? I mean, yeah, even if it wasn't wet, the thallus doesn't look right for Schwanitziae. But Schwanitziae does have those the shape of the epithelium that can be a little bit more kind Globular.
Yeah, how do you describe that? Not uniform, lumpy, something like that. Like this one's got like a star pattern coming off of it.
Yeah. Asymmetrical. Yeah, usually Basidia schweinitzii has. I mean, if it was, that bark kind of looks wet. Did you say that was this after a rain? Was it kind of wet?
It was snowing,
Yeah.
so might have messed the. I would say the. And would you call these parathecia or apathecia? They're not quite parabola, right?
I think they're apathecia.
Okay, cool. We'll keep looking at it, but you think, and am I saying that what right? Puelia?
Yeah, let's start with Buellia. I mean, those things you really need to look at spores anyway. So
Okay, cool. Well, thanks. I've been uploading a lot of things that I'm stopping at ASCO my seats recently, so appreciate the presentation. That was good.
yeah. All right, Christopher, you got one to share?
Yeah, I got one. Hey, Brandon, that mystery bark before.
Yeah.
I was looking at pictures of bark in my tree book. Downy serviceberry.
Downy serviceberry. All right. I'll look it up. I'm sure I'm going to have to wait till like spring to figure out what that tree is, but I appreciate it.
I just found myself in a rabbit hole there for a minute. All right. I just found this blown out of a tree. And when I looked up, not the whole tree, but the whole very top of the canopy had a whole bunch of this. And I tried to get decent photos. The last time somebody showed one of these, you were looking at like just the tips of them. So I tried to get as good at that as I could. And then I even sliced one open a bit.
Cool.
Which those things, when I was scraping away at it, they're surprisingly hardy.
Yeah, they are, especially when they're dry.
So is this exposed area? Is that the medulla, or did he scrape too much? And we're looking at the interior, not the medulla any longer.
Well, with these guys, the medulla is going to be pretty much the entire inside. So, um, you could even take some scissors and clip the end and look kind of down it lengthwise, and you would see the entire interior is going to be the medulla, and then there's a thin cortex around it.
Ah, okay. Thank you. So when I've been trying to look for the medulla and going real light, I think I might have been looking for the cortex instead of the medulla.
Well, the cortex is what's visible on the outside.
Okay. All right.
Usually, I mean, these things, when they're wet, they can kind of be stringy and stretchy. So usually you can just kind of grab with your fingernail and pull it apart. And it's like a mozzarella stick.
Huh.
Gotcha. So it's just cortex and then medulla, right? There's not a layer in between there.
Correct.
Okay, cool. Thank you.
Yeah, so this one might be Usnea strigosa. That would be my first guess, even though it doesn't have apothecia. It looks like it has a white medulla. So
And on these, like the apothecia, are they like? I could almost swear that they somebody else showed when they were kind of like almost little cups in there.
yeah, or discs,
Discs.
hairy discs. Yeah.
I've never found one like that.
Yeah, they look like little satellite dishes.
Yeah.
Cool.
While we're on the subject of satellite dish uh, reproductive structures, so going back to apothecia versus parathecia, like in my head, is it disc shaped or is it not disc shaped? Can you correct me on that, Jason? Like, is it the difference between the microscopic reproductive structures, or is it that simple?
A parathecia, I think, are more pear shaped than disc shaped. They're um usually they're kind of embedded in the thallus. So they're kind of these openings that kind of emerge through the top, and if you take a cross section, you can see that it's kind of pear shaped. Here, let me see if I can find a. This is a decent.
While Jason's looking for that, I think Paul was the only other one with something to show tonight.
Yeah, we'll do Paul next.
I sent a
Yeah.
few lichens, but I want to go last. But if there's time,
Oh.
I think I sent an email. I mean, it's nothing exciting, but there was one thing that where there's something interesting to mention.
All right, cool.
I think I'm gonna, Paul here, I think I'm gonna hold off. Mine's actually not a lichen. I'd rather just hold off till next week.
Okay. So yeah, this kind of pear-shaped structure here in the parathecium, relative to the more flat apothecium. Usually this can be kind of embedded in the thallus.
Okay, all right. So something like Lecanora, does that have a parathecium or an apothecium?
That's definitely an apothecium.
Okay, all right. Cool. And something like Physia would also be an apothecia?
Yep.
Cool. All right, nice. Thank you. That does help.
All right. Dave, you want to go next?
Okay, yeah, nothing exciting. Just the pins that grow on Tricheptum biforme. Now, I knew this was Tricheptum biforme. Oh no, wait, no, this is the one I don't know. I'm sorry. I knew this was Tricheptum, but I didn't note the substrate for the Tricheptum. So when I posted it on Mushroom Observer, Jason Hollinger suggested that because I didn't know what kind of trichaptum it was, that it could be this other thing, Canotheca.
Balsam conensis.
Yeah, but yeah, so that's the one that grows on Tricheptum abiatum,
Right.
and so I just thought, I didn't know anything about this. I just knew about these pins on Tricheptum. So when I saw them on a Tricheptum, fruit body, I thought, well, it's that usual thing. But anyway, so what's interesting to me is now that they, as you mentioned in your talk, Jason, that this other species that is possibly less common is perhaps more common than we think, because people may not have been distinguishing between the two types of trichaptum that might be the host. So
Right.
the interesting thing to me is that my understanding is Trichaptum abiatum grows on conifer wood. So if you get into an area where there's a lot of conifers, and you find some trichaptum, then that might be a worthwhile thing to look very closely at, because you might be able to find some of this this other species. So I think I'm right here. The one that grows on the Trichaptum biforme, which is supposedly the more common one, that is a non-lichenized parasitic entity, right? So I guess we'd call it a fungi.
Right.
And but the other one is a lichenized entity, isn't it? Or am I wrong?
Correct. Yeah, the one that grows on Abitinum is lichenized.
So there's also some lichen on the. So you would find some algae
Yep.
at the base of it. So that would be one way of possibly being able to distinguish. But also, what I've read, or Jason Hollinger may have actually said something in that in a comment, is the spores are different in these two species. So that's another thing to want to check, I guess. But I couldn't find any information on the spores for the one that grows on abiatum. I guess that genus starts with C, I believe, right?
Right. Canotheca.
Yeah. So I'm not sure what the spores would look like for that, because I couldn't find any description or photo. Now that I don't know, maybe it's in. I didn't look in any books. Maybe it's in Brodo.
Well, here's the paper by Jesse Allen and McMullen that describes Balsam conensis.
Oh, okay.
And here they've got some photos showing
Oh, those are the spores, E and F.
So this is Canotheca ferruginea, and then E is Balsam conensis.
But those spores are a lot different than the other thing, because the other ones where they have like sort of pill more elongated shape. These are like subglobose.
Yeah.
Okay.
And they were septate several times, right? Or was that a different thing? And I'm
The other
misremembering.
ones were septate, I believe, with one septum, I think.
You can see here they did a squash mount in KOH. So this KOH reaction is K plus red in the squash mount.
And would you see that, like, if you hit it with KOH in the field, would you see that, or you got to do that on the scope?
I think you got to do it on the scope.
Yeah, cool. And I also noticed, sorry, Dave, I would like to ask a question. I know you're presenting right now, but I noticed you do a lot of work, Jason, at 100x to kind of get like with a section of the thing. Is it for that reason that you're getting that nice red halo for these chemical reactions and things like that?
That I use.
Yeah, and when I say 100x, I mean at 10x. So 100, not 1,000 magnification.
Usually if I'm taking a photo at 100x, it's because once I go up to 400, the depth of field sometimes makes it difficult to get a clear photo. So sometimes I'm better able to take a photo at 100x and then use the zoom on my cell phone as I take the picture through the microscope.
Gotcha. That
Huh,
makes sense.
I've never tried that.
Yeah, I should try that too.
Yeah,
That makes
that's an interesting idea.
my scope even has a zoom on it, so I should try it with that too. Cool.
Yeah.
Thank you.
All right, Dave, let's see. You got one other one.
Yeah, it's just the one that grows on Biforme.
Mhm.
That this was from New Jersey, actually, from the Haynesville Woods Preserve. But I knew that this was Biforme because it was in a. I think it was probably on poplar, actually, aspen. There's a lot of aspen trees in the lower section of that woods, and I'm pretty sure that's what this was on because I think I might have a photo of the tree. Oh, maybe
Let's
not.
look at the photo you have.
Oh, you have to go to the link. Yeah, you have to go to the link there to see the actual. Um,
Oh yeah.
yeah, yeah. I think this is probably one of those poplars. There's quite a few of them in that lower section, or it might be an oak.
Cool.
And
Yeah, but it was
since
on one of those. So I know because of the substrate, because of the polypore. So I know the substrate was Biforme. So it's this more common non-lichenized entity.
Do you have the distribution for the that, not the tricaptum, but the calicioid that grows on it? Do you have that right there or no?
The lichenized, the Canatheca balsam conensis.
Yeah. Well, the one that grows on the tricaptum biforme that he.
Oh, Biforme.
Yeah, I was just wondering how
Pretty
pretty
widespread.
widespread. Yeah.
That's
Okay.
probably all east of the Rockies. Probably find that almost any place, I would guess.
Yeah.
yeah, and
But-
you're seeing some of the population kind of I don't know similarities there, where it's reported more from high density populated areas. I'm sure it's pretty common everywhere. Yeah, east of the
Right.
Rockies.
Yeah,
It
I'd
looks
say so.
it looks that way. Yeah, I looked for it. I didn't see it on the tricaptum, so I'll keep looking. I just wanted to make sure it was worth my time here. So good. Thanks.
Yeah, I mean down in the southeastern part of the US, you probably get more pines and conifers, so it might make sense that there'd be a lower density of the species that grows on a uh, uh, hardwood species, but then out here you get some prairies, but certainly in the forested areas.
I want to find that.
Yeah, Jason will tell you he finds it all the time. I do not have that same result. I look at every tricaptan because I was going to ask, like, where do people here stand with the current, you can't call them all Biforme anymore, I think.
Mhm.
But is that maybe a thing as well? Does it only grow on the true Biforme or whatever we have that's more popular here or more abundant here?
Right.
Yeah, that's a good question because I think there's a tricaptum that grows on poplar that's not Biforme.
Yeah, I've been trying to get my head around them, and like I said, I'm really bad at identifying my trees in the middle of winter. I'm having a tough time with it, but I'm finding a ton of tricaptum everywhere I go. It's super common, but what do you call it at this point, except for Biforme group, and go from there.
There's a good winter tree ID guide that focuses on bark. It's a bark ID field guide that you might find useful.
Yeah, that sounds good because it's always there. Yeah.
Yeah, the problem with bark is though, um, lots of different types of trees have bark that looks radically different when it's young as opposed to when the tree is mature or old growth.
Yeah, so
True.
this field guide has 3 photos for all the species it covers of the young, middle-aged, and old bark. And it
Oh,
has
okay, well, that
photos
would be
of
really,
all of
really
them.
useful.
Which one is it?
Let's see, just Google bark field ID guide. Here's the first one that comes up. Bark field guide to the trees in the Northeast. This is it.
Ah, cool. That looks awesome. Yeah.
Yeah, so here's a young, mature. You always have a quarter in here for scale, distribution, shape of the leaf.
Yeah, that's good.
I mean, like, yeah, there's some trees, you can just kind of, the poplars you could stare up at and see all the pods that are still on there,
Yeah.
or the sassafrasa. Sometimes they still just like have that green tinge to them. Or the yellow birch, it's got the real furrowed old bark at the bottom, but if you look at the branches, it's got that like young tree bark.
Is it beech trees that like rare like lose their leaves really late, or
Yeah.
beech
Called beech and oak both have leaves that kind of
linger?
discolor but stay attached to the branch. There's a term for that called marquescent. They're called marquescent leaves.
Okay, and I would say beech like way more and way longer than oak. I mean, eventually those oaks drop them, but it seems like the beech, I mean, kind of keep them.
I saw a young beech this week with all of its golden leaves and still attached, hadn't dropped.
Yeah, we don't really have them, but in down south, I saw so many, and all the other trees were missing their leaves. It's interesting. Helpful.
You don't have any beach there where you are?
No, not really. No. Mm-mm. Nope. Very rare here. Up north and south of us, but not here.
Hey, it's Hunter. You wanna go next?
Sure. Yeah, if there's anyone else who wants to go, is there anyone else left?
I don't think so. Is anybody else? Check the chat. I think you're the last one.
Okay. Yeah, we can just kind of blow through these, but I really don't know what any of them are. But I'm hoping they're actually lichens. So yeah, this is Northern Illinois, November, October, something like that. Probably don't even know what wood it's on. It could be a variety of hardwoods. But anyone know what it is?
Looks Hydnoporia.
Oh,
Yeah, I don't think this is a lichen.
oh,
Although I see a lichen here.
Yeah, there is lichen in the photo.
I see one here. I see at least 3 different lichen species. But yeah, this looks like Hydnoporia.
oh, okay. I don't really know that. So that's cool.
Did you find this cool? What's going on there? I was going to ask a question about the white tips, but if you go to that next photo, there's a white, and on the left side here, there's a white thing kind of moving in.
Yeah, there's a lot going on in the next photo, but I have no idea what. And those black globules are kind of interesting too.
That's a lot.
Yeah.
I would say so. My very loose guess would be this is Hydnoporia olivaceae, and there is some sap that moved in.
Okay. Can
Yeah.
someone put that in the chat for me, the spelling?
Yeah, I got you.
Thank you.
Was this stick right side up or rolled over?
This is how I found it, but it may have moved.
So here the white tips look more yellow. Is it possible these are spores?
Hmm. Yeah, kind of pretty. Thank you.
Yeah, that's a possibility, Jason. You're talking that on those kind of clubbed tips.
Yeah.
So the Hydnoporia has a few different forms, or
Well, if you actually zoom in kind of dead center at the top here. Yeah, just to the right here, you can see the telltale Hydnoporia thing where it's just those elongated kind of angled tubes kind of
okay.
towards the apex a little bit here or towards the top. Yeah. That one right there, the one next to it. Yep. It's just kind of like a hollow, elongated pore.
Interesting.
Oh, so Hydnoporia, like poor
Tooth pore.
teeth?
Yeah, tooth pore.
Oh, man.
Interesting. Okay. Yeah. The color changes on there are kind of neat, but yeah.
Yeah, that species Olivacea in particular, wildly variable, depending on like the hydration levels recently and how old it is, maturity level, and all that. Because when they're fresh, it really does feel kind of feathered and, yeah, like really light and feathery. And then once they dry out, um, they're no longer feathery. They're kind of sharp.
Yeah, when I've seen as I looked at the pictures of other specimens, they looked sharp. It's interesting. And then the black those black structures is something different.
Yep. I think that's something different, but I don't know what. I don't think it's a lichen.
Okay. All right.
I don't really see much of a thallus underneath it. It looks like it's coming straight out of the bark.
Yeah.
But this foliose lichen down here in the bottom left, that looks like a Physia.
Aphysia. Okay. Pretty.
A member of the genus Physia. The genus starts with a P, not an A.
Oh, okay, thanks.
And then this is some type of green sterile crust. Several lichens that generally look like this without fruiting structures can be challenging to identify.
Cool. Okay. Good. Oh, yeah. Are these lichens?
Yeah.
Yep.
Okay.
Probably Physia.
Okay.
It's hard to tell without seeing a closer shot. But
I did have one, but I didn't include it.
Most of this, there's kind of this gray lichen down here, and then some of this black is probably also a lichen. This might be a different lichen.
Mm.
Oops, some Uzia.
Oh, look at that on the ground there.
Yeah.
Neat.
Some other stuff on those trees on the left there, maybe.
Yep.
It's a lot of Spanish moss, I think.
This is down south. Huh?
Yeah,
Oh yeah, look at it. Oh,
yeah,
there.
yeah. This is Louisiana. This is back in Illinois. It's really pretty. All the shades of blue and green. And
Yeah, so you see all these little white dots, the thallus.
mm,
That's a good ID feature. It's not the only species that has white dots, but it's probably the most common. This is Punctilia rudecta.
mm.
Just to check.
PUP.
PUNC. Just put it in the chat.
Oh, thank you. Yeah, this one was really pretty. Oh, and I showed this fungus before, and then I was noticing all this cool stuff in the photo that so I went back and cropped it so we could look at that. That looks a lot like what Jason had.
Brandon had.
I mean Brandon.
Yeah.
Yeah, those black blobs. Yeah, similar.
Yeah, they're kind of globular in here, except you can see kind of one that's got more of a stereotypical rim that you would see with a flatter apothecia. So you see some black rim here too.
And then the ones on top is that what you meant by flatter or no? That's different.
Yeah, so this one's definitely a Lechinora or something more a Lechinoran, just the adjective to describe the species. Probably something in the genus Lechinora. It's different than this guy down here, which is probably a Buellia. You got a different species here. Brown apothecia.
Exciting.
You can see this zone.
Mm.
Forget what it's called, the zone here where the one lichen meets the other and they're emitting chemical defenses as a way to kind of compete.
Right. Interesting.
But you could even see between individuals of the same species. I mean, here you've got a Lechinora versus what probably a Buellia, but sometimes you can have a Buellia next to a member of the same species, and you'll also see this zone where the two thalli meet.
Very cool.
And Jason, does that Buellia look more kind of typical for the genus? Maybe one of the, I forget the name of the common one that we get, but does that look more typical?
Yeah, I mean, you can see here you've got some, some of these have that kind of stereotypical rim that you would see. Looks like some of these are trying to be more, flat apothecia like rather than the globular masses that yours had. But I still think they're both Buellia. That's my initial guess.
And I just came back in. Did we discuss that big polypore, Trametes?
No, not yet.
Oh, we did. No, we did last time or the time before. It's
Oh, okay. Cool.
yeah, yeah. It's the
Lactania.
yeah. Wait, what did you say?
I said.
It's Lindsay's.
Oh, okay.
Yeah,
Cool.
I mean, that's what we said it was. So, right.
So it has gill. This one has gills.
Right. Yes. Yeah. It definitely does. The only other thing that I thought after the fact it could be was the Daedalus confrugosa. But otherwise it's what the Lenzites
Yeah, there's one Daedaleopsis that actually has hymenium that's more like gills.
yeah.
It's sort of an uncommon one. Confrugosa usually just has elongated and maze-like pores,
Oh,
and
okay.
if you rub the pore surface, it'll stain reddish brown. But there's
Okay.
another Daedaleopsis that really can be confused with
It's Trimides.
Trimides betulina, which
Yeah, Hunter.
used to be
Just real quick.
Lenzites. Yeah, used to be Lenzites.
Oh,
You're
it's
on it.
Trinity's now. Oh, okay. Thank
Yeah.
you. Thank you.
I think this other Daedaleopsis is I think it's host specific on birch. I'm not sure. I can find out in a minute.
Okay. Yeah, that must have been what I was thinking it looked similar to, I think. But anyways, I was just showing the picture for the lichen. So this time. Is that the last one?
Yeah, that was the last one.
Oh, okay. Okay. You do it about it. Jason, that was such a good presentation. It was
Thanks.
really neat. And the is it multiclavulina that is not in that group?
Right, because that's not, that has tiny stalks, but it doesn't have a capitulum at the top.
Oh,
Can
okay.
can you describe a capitulum? Like what how does that differ from an apothecia or a perithecia? Is there an outer membrane that kind of dissipates or what?
It's just shaped like a head.
Yeah.
Pinhead.
Decapitate.
Okay, cool.
Oh,
I put the other Daedaleopsis into the chat. Septentrionalis. It grows on birch, like I said.
great.
Yeah, it can be confused with Trimides betulina. They look kind of similar.
Yes. Yeah, because I thought it was the Trimides betulina, but then I was like saw the Dadiolapsis and was like, oh, maybe I made a mistake there, but I think not. But thank you for sharing that.
Okay, you're
Yeah.
welcome.
Yeah, and just on that Septentrialis, the Daedaleopsis versus Trimides betulina, they're saying that the gills, quote unquote, right? The pore gills, way tighter, if I'm not mistaken.
Oh, tighter on the Daedaleopsis?
Yeah,
I think so. Yeah, I think that might be one way to
and
get
shallower,
an idea about the difference. Yeah.
and shallower on mature fruiting bodies. If you have a mature fruiting body, I'm pretty sure Trimides betulina gets like those what do you call it? Yeah, gills. The false blades are longer in length.
Oh, like wider. I think that this Daedaliopsis thing, I think the gill-like things get to be pretty wide. Let me just check on that because I think I have that website ready to look at in the other window.
It's good to talk about because these things can be really confusing.
Yeah.
You know, even the maze ones that are supposed to be host specific. You know, if you find one on oak, it should be quercina, right? Or quercina.
Right.
Daedalia, of
Yeah,
course,
right.
yeah. That one has really thick pore walls.
And yeah, and it's like real maze. Well,
Yeah, and it looks really mazy. Yeah,
maze like.
mazy.
But even that one can be confused for you know, some species of Tremete things that are still in Tremetes, and some of the non-oak associated Daedalea type things. But yeah, that's all I'm saying.
Well,
Yeah,
actually,
things look so similar sometimes, and yeah, I'm yeah, just constantly amazed at at that.
Yeah, I feel like I'm always, oh yeah, you can tell these things apart because of the things that I've read, right? And then I get in the field and you're like, wait a minute, that's halfway between these two things. Give me a break, you know?
Yeah, yeah. I mean, more and more, it's like the less it's like the more you know, the less you know. Actually,
Yeah,
actually.
agreed.
Yeah, the
Yeah.
less confident you are about some of these crazy things. Yeah.
Yeah.
so the Michael Quebec says about Daedaliopsis septentrionalis, blades, thin, wavy, up to 2.5 centimeters tall at the base. So I think they're talking about the width, actually, of the
Oh,
gills. So they can get so that when they get
That's
they can
huge.
become rather yeah. So then, but it does look to me like they're probably a lot closer together than the typical Tremete spatulina. I'm not sure. I think there's a difference in spore size. Anyway, yeah, you get in the field, and I know what you're talking about. You find these things, and you look at them and say, I'm not sure what this is." Happens to me all the time.
Yeah, I mean,
yeah.
how many times have we had this conversation, right? Talking about Daedaliopsis, Daedalia, Tremetes, whether it's the guild ones or it's the maze-like ones, and you know, we come away with like, yeah, I think that's a consensus, and then you get in the field and you start reading another guide, and you're like, nah, it's all out the window.
Yeah, but yeah, I've been going through my photos from like years ago and things I like slapped a name on. I was like, no, I'm like, I think that's not right. Like that, I don't know what I was thinking. Like that is not right. Yeah.